I welcome you all back to our YouTube channel Ignited Medicals. So today's class will be about the urea cycle and I am Manoj. So in the university examination they will give this urea cycle as a 10 marker long essay question. So in which the question will be asked as a describe the detoxification of ammonia by the urea cycle, explain its regulation and disorders. So it's a long essay question. So now the urea cycle, it's also called as a Krebs-Hanslett cycle and ornithine cycle. It's called as an ornithine cycle because the first substrate for the urea cycle is ornithine. So it is called as an ornithine cycle, and Krebs-Hanslett based on the biochemist who have discovered it. So as we all know that urea is the end product of the protein metabolism. So whatever the amino acids are present, it will be converted to, at the catabolic reaction, it will be converted to carbon skeleton and ammonia. This carbon skeleton will undergo the oxidation to produce the ATP and we'll get the energy from it. And the toxic compound is the ammonia. This has to be converted into urea and it has to be eliminated from our body. So now this ammonia has to get detoxified. So this detoxification of ammonia will be carried by 3 steps. That is reduced production, ammonia trapping and urea synthesis. And the carbon skeleton, it is not our concept of discussion for today. We are majorly focusing on ammonia. Right. Here the ammonia is trapped in the form of glutamine. So the ammonia will be trapped in the form of glutamine. How this glutamine is formed? The glutamic acid will combine with this ammonia to form the glutamine. Okay, the ammonia will be trapped in this glutamine. Further, during the urea synthesis, this glutamine will split into glutamic acid and it will release the ammonium ion. This glutamine will split and it will release the ammonium ion which enter into this ornithine cycle. So now let us focus on the ornithine cycle. This ornithine cycle consists of the 5 steps. So in which you have to remember all these 5 steps with the enzymes, and each enzyme will deficiency is one of the inborn error. So by remembering this enzyme name, we can easily remember all the defects, the inborn defects of the urea cycle. Here in this first step, that is the ammonium ion which is released from the glutamine, it combines with carbon dioxide in presence of carbamyl phosphate synthase 1. So you can remember this enzyme name based on the substrate. Carbon dioxide, amino, phosphate means here phosphate is obtained from this ATP. So the name is carbylamyl phosphate synthase 1, which will produce carbylamyl phosphate. The second step here is the formation of citrulline. So the ornithine is the first substrate of the urea cycle. Okay, this ornithine will combine with carbamyl phosphate in presence of the enzyme ornithine transcarbamylase. Ornithine transcarbamylase will convert into citrulline, will form the citrulline. So this is the formation of citrulline and the second step. The next step is the formation of Arginosaccinic. The citrulline now will combine with aspartate. Will combine with aspartate in presence of Arginosaccinic synthase. Okay, the formation is Arginosuccinate. So the enzyme is Arginosuccinate synthase. Will form the Arginosuccinate. Here ATP is converted into AMP, so which there will be 2 ATPs involved in this reaction, right? Yes, and in the first step there are 2 ATPs involved, and here there are 2 ATPs involved. So remember this point. And in the next step, that is the formation of arginine. This is arginine. So now this arginosuccinate will undergo lyation, means splitting reaction, in presence of arginosuccinase or arginosuccinolase. Arginosuccinase or arginosuccinolase will form. You split this arginosuccinate, you get arginine and fumarate. Arginine and fumarate. Yeah. Our last step is the formation of urea. So how urea is formed? Now this arginine, we have to hydrolyze the arginine. Okay. In the last step we got arginine and fumarate. Fumarate is, it is involved in the further cycle. I will tell you the relationship. Now this arginine will undergo hydration, and in presence of arginase it will give the urea, and ornithine is obtained back or regenerated back, which will be involving in the other next urea cycle. So now this is what we get the urea. Now let us see this cycle in detail. Yeah, see now this is the first step, that is the carbon dioxide combines with ammonium ion in presence of carbamyl phosphate. Synthase 1 will form the carbamyl phosphate, and now this carbamyl phosphate will combine with the ornithine. It will form the citrulline by the help of the enzyme ornithine transcarbamylase. Next, citrulline will convert to arginosuccinate by combining with aspartate. Citrulline combines with aspartate, and in presence of arginosuccinate synthase, that is arginosuccinate synthase, will form the arginosuccinate by utilizing the ATP, and further this arginosuccinate converts into arginine and it liberates the fumarate. Okay, it will form the arginine and fumarate is released. Okay, now this arginine will undergo the hydration, okay, in presence of arginase enzyme, and it will release the urea, and urea cycle is completed by regenerating this ornithine, which participate in the further urea cycle. So the first 2 reactions will be carried out in the mitochondria, and the remaining steps will be carried out in the cytosol. Now this is all about the urea cycle. Now there is some relation between the Krebs cycle and the urea cycle. So what is that relationship? Means we already know that. What is that? The carbon dioxide and the ATPs used in the urea cycle will be obtained from the citric acid cycle. And it will be used in the urea cycle. In the same way, the fumarate, here there is a liberation of fumarate in this step. It will be used in the citric acid cycle. Clear? Yeah, the cycle is incomplete without looking into the energetics. Let us have a look. Yeah, we already know from the urea cycle that there are 2 ATPs are consumed in the first step, and two more ATPs are consumed in the third step, right? So now totally there are 4 ATPs are consumed in the urea cycle, and urea cycle forms 3 ATP. Okay, why? How these 3 ATPs are formed? The fumarate, which is released in the 4th step, enters citric acid cycle, and this fumarate generates 3 ATP. So the net utilization is minus 1 ATP. So there will be 1 ATP utilized in the urea cycle as a net. So the urea cycle consumes 4 ATPs. It generates 3 ATPs via fumarate, and the net utilization is going to be the 1 ATP. Yeah, now let us move on to the regulatory part of the urea cycle. The, the regulation of urea cycle is at the first step, that is carbamyl phosphate synthase 1 is the regulating enzyme in the urea synthesis. So this carbamyl phosphate synthase 1 is allosterically activated by the N-acetyl glutamate, that is NAG. Okay, for the activation of this CPS1 we need NAG. Without NAG this is not activated. So therefore this NAG is synthesized from the acetyl CoA and glutamate, and when the amino acid catabolism is increased, the concentration of this glutamate will increase. When this concentration of glutamate is increased, obviously there is an increase in the production of NAG. When there is an increase, at this time the CPS is maintained at the normal condition. So this is the method of the regulation of the urea cycle. It's mainly by the CPS1. So now let's move on to the defects. When defects occur, what are the things that are possible? Yeah, as I already mentioned, there are 5 steps, there are 5 defects. 5 steps, 5 enzymes, 5 defects. So the first enzyme is carbamyl phosphate synthase 1. When this is defected, so what occurs? It causes the hyperaminomia type 1. Hyperaminemia type 1. So what are the characters? So there is a deficiency of carbamyl phosphate synthase 1, means the first step will not occur. When first step will not occur, what happens? There is an increase in the ammonia content in the blood, and it can lead to the mental retardation. So that is the characteristics of hyperaminemia type 1. Next, hyperaminemia type 2. It occurs due to the deficiency of the ornithine transcarbamylase. This is a step 2 enzyme. So when this is deficient, it causes the hyperaminemia type 2. So what is the substrate in this reaction? That is the ornithine and carbamyl phosphate. So when this is defected, what happens? There is an increase of ammonia in the blood and glutamine. Ammonia and glutamine. The next is citrullinemia. Citrullinemia is caused due to the deficiency of arginosaccinic synthase. This is the third step, third enzyme. So now here there is characteristics are increased ammonia and citrulline, because citrulline is not converted into the arginosuccinate. So therefore there is an accumulation of citrulline. Next is Arginosaccinic Acidoria. This is due to the deficiency of arginosuccinase. So the arginosuccinate is not converted into as arginine, right? So that what happens here? Arginosuccinate concentration increases in the blood as well as the urine. And the last is hyperarginemia. Okay, hyperarginemia. It is due to the deficiency of arginase enzyme, and it increases arginine concentration in the blood. So this is the very easy to remember. So this is all about today's class. So please do like, share and subscribe our YouTube channel.
Yeah, thank you all.